database looking for analytes in low abundances. So now that we have a starting material, we selected the simulation conditions for our experiments. We were particularly interested in investigating conditions near to the ancient Martian geothermal gradient, especially as they approach hydrothermal conditions, as you can see represented on this temperature pressure diagram. And for these experiments, we selected 3 different compositions of fluids: pure water, a simple sodium chloride brine, and then an acidic brine with different ratios of magnesium chloride, sodium chloride, and potassium chloride, according to model results for a late-stage brine formed from basaltic weathering, fluid evaporation on Mars. So then these materials were subjected to a variety of different pressure and temperature experiments. The principal experimental vessel we used was these batch reaction vessels, which can accommodate up to about 750 milliliters of fluid. And you can see them sitting in the furnace here with the rock cores beneath them. And these go up to about 15 megapascals of pressure and about 80 degrees Celsius. We also use the piston cylinder press for an anhydrous experiment comparison. And it has a really wide pressure and temperature range, up to 4 gigapascals of pressure and 2000 degrees Celsius. And then finally, we used an ambient pressure furnace just for an ambient pressure comparison. So only looking at the temperature there. After the experiments, we did a variety of different sample characterization. Of course, we were principally interested in organic analysis, where we used GC pyrolysis for bulk sample characterization. We used liquid injection GCMS and also LC-MS, where LC and liquid injection GC were particularly suited for trace analysis, where the analytes were in lower abundances. Naturally, this involved some sample prep for the liquid injection samples, including extraction, separation by polarity, and then different derivations depending on which compounds we were interested in. And you can see a few of the leftover salts there from the extraction process. We also did a variety of different inorganic analysis techniques, such as XRD for monitoring the mineralogy of the samples and their mineralogical degradation under higher pressures and temperatures. And then also ICP-MS for the fluid composition following our brine experiments. So here are some results of that instrumental analysis, where you can see that there are abundant peaks in both the liquid injection GCMS starting material and the LCMS starting material. So the samples are very bioorganic rich as expected. And most of the compounds that we're interested in were above the detection limit for all of the instrumentation that we wanted to use. We were able to identify many of the compounds by GCMS database, even when we didn't have standards. So before I discuss the results from the brine experiments, I first want to show you some results from another study in an anhydrous system, where we were determining the pressure and temperature effects on biosignature degradation. In the light blue there, you can see the untreated sample analyzed by pyrolysis, where there are again abundant peaks. Many different bioorganic compounds can be seen. And then just beneath it is the piston cylinder experiment run at 8 kilobars and 300 degrees Celsius for 8 days. And even there, under those high temperatures and that high pressure, you can still see that there are a number of discernible peaks. You can still identify many different degradation products, and then even some retained original compounds. It isn't until you get to 550 degrees Celsius that we see really extreme, severe degradation of the sample, where you're starting to approach that blank baseline and nothing can be discerned. So in this way, we had an idea about the degradation mechanisms of these samples and the preservation potential of the different compounds within it. However, this was for an anhydrous system, so we were really interested in whether or not a fluid-rich environment would change our original interpretation of pressure and temperature effects. It is also worth noting that we ran these both by pyrolysis and by liquid injection. And more or less, we were obtaining the same results, even though these different techniques might have different sensitivities to different analytes. We could still see very clear starting material signatures and very clear different degradation effects. So, how does this compare to the brine samples? Well, this is where things become a little bit more unexpected. So when we first ran the LCMS samples, we noticed that all of the peaks were starting to look the same. Whereas before we could see very, very clear temperature and pressure effects. Now suddenly the low and high pressure samples, basically for all the different compositions, look more or less the same. Furthermore, when we ran these samples on the GC, everything was beneath the limit of detection. We were obtaining flat baselines. So already these samples from the same starting material are behaving really differently. And it is worth noting we were able to obtain the rock cores pretty much more or less intact. What was looking as we expected was ICP fluid analysis, where sure enough, the acid samples showed some dissolution of the carbonate, where you can see that indicated by the higher calcium counts. And it turns out the organics are where you would expect them to be. So we took those enormous volume 750 milliliters of liquid, and we reduced it down and analyzed those. And this is where we see those missing organics, which tells us that there's clear partitioning of the organics between the solids and between those brine solutions. Furthermore, it tells us that those organics are highly mobile in the fluids, which isn't something that you would expect for all of the compounds, particularly the ones that are not easily extracted in water. And also, finally, we're seeing some unique peaks based on the fluid chemistry here. So the samples don't look all the same, which is a relief. It's also worth noting that we're really approaching the limits of detection of this instrumentation. So this type of analysis necessitated a really careful characterization of all the blanks, the reagents, and then of course running instrument blanks throughout the entire process to ascertain whether or not these peaks were legitimate in the samples. So, as a whole, what we learned from this study was that biosignatures behave dramatically differently in these brine environments, in these fluid environments, compared to non-nominally anhydrous environments, where many different compounds were highly mobile in the fluid, even when the structural integrity of the core was maintained. So this might indicate that if you're looking for biosignatures in a sample, if they were experiencing this kind of fluid process, they might be leached out, they might be moved somewhere else. And then we did see that the different fluids provided different products. And whether that's due to the carbonate dissolution, the sample actually dissolving, or if it's due to different acid catalyzed reactions is something we're still interested in. Namely, we're looking more into deconvoluting the LCMS data, which is not a trivial task when you're doing this kind of exploratory analysis. And that's going to let us elucidate some of these specific reaction mechanisms and pathways. And I'd also like to do further analytical work on these samples, such as for the aromatics by UV vis. All right, well, thank you so much for listening to my talk. If you have any questions, I'd love to hear them now, and you can contact me here.
Thank you, Ardith. What a great talk. I have a question if there's none coming through the group chat. Oh, here we go. Erica says, it would be interesting to see what happens when with different geological mediums, chert, silica, quartz, shale, etc. That actually is along the same lines as my question, because of course organics behave quite differently with carbonates than others, and you usually would dissolve carbonate to free bitumen 2, for example. So you might be having some artifacts from the dissolution of the carbonate. Is there any plans to try this with other geological mediums?
Yes, I really love this question because this is a research project we're currently in the middle of working on. And I completely agree about the carbonate dissolution too with the acid. That was something that we could clearly discern there, even if the entire carbonate wasn't compromised. But back to the original question, I definitely think that there's going to be some really important matrix effects and mineral organic interactions. I see that we have tart, silica, quartz, shell, etc. I did mention we had some lovely shell samples that I would love to core and look at. Currently, we're trying to work with clay right now, which is a bit of a challenge because you can't really core it and put it in these batch reaction vessels. So there's a bit of an experimental limitation here, but certainly something we're interested in.
That's great. So we're out of time for your talk, Ardith, but I know I will be in contact with you by email to continue this discussion further. So thank you so much for your talk. That was wonderful. And our next talk will be from Michael Macey. So Michael will be talking about the identification of sulfide oxidation as a potential metabolism driving primary production on Noachian Mars. So Michael, if you're there, and you'd like to
Yes, yeah, I'm here.
sharing. Beautiful.
Fantastic, and I will hit start presentation. And that button won't work. So
Fantastic, thank you.
thank you. So thank you very much to the organisers for this opportunity to present on a recent collaboration between us at Astrobiology OU, based at the Open University, and Claire Cousins' research group at the University of St Andrews in Scotland. So this work was characterising an analogue environment. So for the purpose of this work, we're defining an analogue environment as a region possessing a chemical environment approximating that of a celestial body of interest. Approximating because we do not have absolute certainty of some of these values from these celestial bodies, but also because on Earth, even in anaerobic environments, there will be the presence of minerals and chemical species formed in an oxygen-rich atmosphere. So for example, an analogue for the surface of Mars today would be saline, oxidizing, anaerobic, and exposed to high levels of ultraviolet radiation. Whereas an analogue environment for the icy moons would be alkaline and highly saline based on the modeling, exposed to extreme temperature gradients in the interior, and exposed to high levels of radiation in the exterior, which of course brings up the key point that an analogue environment is an analogue for a specific location on a celestial body and at a specific point in its history, with the surface and subsurface of Mars believed to be incredibly different, and Mars 4 billion years ago requiring a distinct analogue from one characterizing Mars today. One such proposed analogue environment is Axel Heiberg Island, which is located in the Canadian High Arctic. It's characterized by polar desert conditions, so it's very cold and dry, and lies within the region of continuous permafrost, with this indicated on the map in white. The red dots on the map indicate a series of sulfur-rich, highly saline and perennially cold springs, which do not freeze in spite of the fact the air temperature is below zero all year long, and the blue dot represents the McGill Arctic Research Station, or MARS, from which all research on the island is launched. We're focusing specifically on one spring, Colour Peak. The springs here are slightly less saline than the neighbouring springs, but they are much more sulfidic and sulfurous, as evidenced by the black coloration of the sediment itself. Now, from the microbiology perspective, this site was of interest because the microbes that survive here have only ever been partially characterized in a study in 2007. However, from the astrobiology perspective, it was interesting because it's also argued to be an analogue for Mars during the late Noachian. Now, when we consider whether something is an analogue environment, the first question is, of course, is it chemically and physically relevant? Well, the air temperature range of Colour Peak reaches -40 degrees centigrade in the winter months and has an average of -20 degrees centigrade throughout the year, which puts it within the range of some models that estimate for a cold and wet Noachian, as opposed to the warm and wet model that is also proposed. We then analysed the chemistry of the water, sediments, and water and sediments in combination from ColourPeak using ICP-OES in order to identify the most abundant elements. As you can see here, these are represented by the red, pink, and black dots in the corner here, with the key point being the neighbouring diamond on the left and right-hand side of the circle. These are thermochemically modelled waters that are derived from the chemistries of Gale crater evaporates developed from the Schwenzer 2016 modelling study, which indicates, at least with regards to the most abundant elements, this is an appropriate analogue with regards to the proposed chemistries of waters during Noachian Mars. So then the question is, okay, if it's physically and chemically appropriate as an analogue site, what microbes are surviving in this environment? In order to answer this, we burst the cells open and sequenced the 16S rRNA gene. This is the universal barcode gene found in all microbial species, which allows us to see which species are present. In the same way, a register tells us who is present in a class. So from extracting the DNA and sequencing this from replicate sediment samples, I don't need you to pay close attention to the diversity of Latin names at the bottom. Instead, I just need you to observe how many of these bars are of purple pinkish shades, with these representing sulfur oxidizing bacteria. And it's clear that sulfur oxidizing bacteria dominates these sediments. Now, why specifically are these of interest? Well, sulfur oxidizers yield energy through the oxidation of reduced sulfur compounds. So sulfide oxidation can occur using oxygen as the terminal electron acceptor, occurring aerobically, but it can also occur anaerobically using nitrate as the terminal electron acceptor, occurring in a denitrification enabled fashion. It's also worth noting that many of these sulfur oxidizing bacteria are also autotrophic, so they convert inorganic carbon, carbon dioxide, to produce their organic carbon. They do not require an external source of organic carbon, as we do as heterotrophic organisms. However, it's problematic to extract DNA from this type of environment in order to attempt to characterize the viable and active microbial community. There are models that estimate that in a cold and saline environment, fragments of DNA that the previous assay we've used would be able to detect would be able to survive for over millennia, meaning that the species profile we develop may be of viable cells genuinely present and also of long dead cells. And this is very important to consider. In order to attempt to overcome this, we elected to also extract RNA and sequence the 16S rRNA gene profile from this, as RNA has a much shorter half-life, even under these preservation enhancing conditions. So comparing a collapsed DNA profile to a collapsed RNA profile, we can see that the level of diversity that's detected within this environment suddenly significantly decreases, with many genera no longer being present. We do, however, observe that sulfur oxidizing bacteria continue to dominate within this environment, with some additional heterotrophic diversity remaining. So the active, or more accurately, viable microbes were also mostly sulfur oxidizing bacteria. So why is this specifically interesting from the astrobiology point of view? From the microbiology point of view, we've certainly gained some level of characterization of who's present in this site and what some of the metabolic processes could potentially be. However, why are we interested that this specific metabolic process might be occurring by some microbes in this obscure cold pool in the high Canadian Arctic? And in a more general sense, why are we interested in characterizing the microbial diversity within analog environments? And the answer to this is they allow for the generation of hypotheses. Now, what does this mean? So if we look at what we know about the ColourPeak environment, we know that it's a carbon poor sediment that's incredibly sulfide rich. and it's dominated by autotrophic sulfur oxidizing bacteria. So sulfur oxidizing bacteria are probably surviving in this environment by fixing carbon dioxide and through the oxidation of sulfide and the other reduced sulfurous species within the sediment. This is something that's also observed within hydrothermal vents, which have a high sulfide concentration. However, as these cells are growing, they will be releasing organic carbon intentionally to modify the environment or accidentally through leakage. Also, as these cells are growing, some cells will be dying, again representing another pool of organic carbon that they themselves cannot utilize. And we know that there are other heterotrophic bacteria surviving within this system. So it's possible that these are surviving partially due to the input of carbon by the autotrophic members of the community. Again, this is something that's observed in hydrothermal vents, but is also observed in some pure culture studies where microbial species are grown together and in isolation. Now we can propose this as a system of carbon cycling in Colour Peak. And we know the concentrations of nitrogenous species, we know the concentration of the relevant gases in the atmosphere, and we know the concentrations of sulfide. But we also know some of these parameters for the proposed Noachian waters. We know the concentration of nitrates that were detected and announced in the Stern et al. 2015 paper by the Mars Curiosity rover. We know that sulfide and sulfates are incredibly widespread and abundant on the surface of Mars and are also contained in Martian meteoritics. And we have estimations for what the concentrations of these individual chemical species would be within these proposed Noachian waters. So the question then becomes, assuming all other things are equal, would this process yield sufficient energy to enable the replication of sulfur oxidizing bacteria? And we can assess this using Gibbs energy equations, which assesses this thermodynamic feasibility. And the answer is yes, under both aerobic and anaerobic sulfur oxidation, they are thermodynamically feasible to yield energy enabling replication. Now, these calculations produce low numbers of cells, but they produce numbers of cells nonetheless. And this is an interesting result for two key reasons. One, sulfur oxidizing bacteria have previously been overlooked as a putative viable metabolism under Martian conditions, and they have the potential to produce unique and specific biosignatures. But also, when we assess the viability of organisms, these are commonly assessed in isolation from each other, and it's possible that we instead need to start considering community-dependent metabolisms, those that can only grow when others are present. So, just to start to wrap things up and ending on some results we've recently got in, the question is, are the microbes that are growing under what we've argued is a proxy for the Martian chemical environment then capable of growth under simulated Martian conditions, more chemically accurate and controlled? And do we continue to see the thriving of sulfur oxidizing bacteria? So we again here have the DNA and RNA profiles of the community, with the purple representing the dominant sulfur oxidizer.
2 minutes left.
Thank you. And the answer that we see is yes, but with distinct communities selected between the simulated chemistries. Under one, simulating that of rock nest at Gale Crater, we see the continued persistence and thriving of sulfur oxidizing bacteria and the co-occurrence of heterotrophic diversity. Whereas in hematite slope, a more iron-enriched simulated chemistry, we instead observe the complete replacement of sulfur oxidizing bacteria with sulfate-reducing bacteria, who were an extreme minority in the original community, thus requiring further analysis to assess whether this is a result of competitive exclusion, chemical exclusion as they cannot grow under this defined condition, or is it simply stochastic variation between enrichments? So in conclusion, the physico-chemical conditions of Colour Peak identify it as an appropriate analogue for Mars during the late Noachian, and it's an environment dominated by diversity associated with sulfur oxidation, a metabolic process that Gibbs energy equations indicate would be thermodynamically feasible under proposed Martian conditions, with future work being the continued analysis of simulation experiments, assessing the viability of the Colour Peak community and sulfur oxidizers under Martian chemical conditions. I'd like to thank again our collaborators, specifically my summer student, James Oliver from Edge Hill University, who is a budding astrobiologist and is also currently looking for a PhD project. Thank you very much.
That's awesome. Thank you, Michael. Unfortunately, we don't have time to answer any of the questions, but there was a question for you in the chat if you do want to answer that. Our next talk will be Yuanyuan He. Are you here? If you are, could you begin to, oh, perfect, you are. So if you want to share your screen, Yuanyuan will be giving a talk about the search for life signatures on Mars, the detection of DNA with TMAH, Thermochemolysis on SAM and MOMA.
Okay. Okay. Hello, good afternoon, everyone. I'm Yuanyuan. I'm a third-year PhD student from St. Helens Belek. And today, my topic, we are going to talk about the search for life biosignatures on Mars, mainly about the detection of DNA or RNA with TMAH thermochemolysis on SAM. And SAM is a sample analysis instrument on board Curiosity, and also MOMA is the Mars Organic Molecular Analyzer will be on board ExtraMars. And for SAM and MOMA, their main goals are to search for life biomarkers on Mars. And these life biomarkers could be carbohydrates, lipids, proteins, and nucleic acids. And in order to analyze the potential organics on Mars, the derivatization method have been used. Here we can say on SAM, there are two kinds of derivatization reagents. One is MTBS, TFA, and DMF, and another one is that is TMAH. And also for MTBS, TFA, and DMF, it has been applied to analyze the organics on Mars. And also TMAH, thermochemolysis, will be applied to analyze the organics in Martian soil also. But here we mainly talk about the lab research about TMAH thermochemolysis. So what's thermochemolysis? We know for organic compounds, there are some non-volatile compounds. This means these non-volatile compounds can't be detected by our device, but when we deal with these non-volatile compounds with our derivatization reagent, they can become volatile, so they can be detected by our device. And this is this process like when the worm when they have wings, they can become a butterfly, so it can be seen very easily. Here we give some example. As we mentioned before, for MTBS, TFA, it's a ciliation, it's kind of ciliation reagent, and for TMAH, it's a methylation reagent. Here, when we analyze this derivatization reagent, their functional group will replace the polar hydrogen. Here we give the example is fatty acids. We'll replace the hydrogen here and form the fatty acids derivative. And during this process, the derivatization will be increase the volatility and reduce the polarity of the organic compounds. From here, we can say that actually the thermochemolysis reagent is very important for our to search for life biomarkers on Mars. And when we mention looking for Mars life biomarkers, the first thing we came to mind maybe is DNA or RNA, and we know DNA or RNA are very important for our life. And maybe in biology, when we analyze DNA or RNA, the first thing we need to extract the DNA or RNA from the real life from their cells. So I wondered if it's possible to detect DNA or RNA with our TMAH thermochemolysis. We only use our TMAH thermochemolysis. So in our study here, our main goal is to test if it's possible to detect DNA or RNA, the life biosignatures from real life with only with our TMAH thermochemolysis. And to achieve this goal, we start from the simple sample. Here, we use our TMAH thermochemolysis, analyze the nucleobases, nucleosides, and nucleotides, and then we tried a little bit complicated polymers. Here we choose poly-A tail because poly-A is very important for message RNA. And last, we analyze the real life with our TMAH thermochemolysis. Here we choose E. coli because E. coli is very common and very easier to get in our lab. Here, this is the this is the planning of our whole study here, and all the experiments have been done with PYGCMS, and the PYGCMS was used to mimic the SAM conditions on curiosity. Here we only give some results. We take adenine as an example. When we analyze adenine with TMAH, we know that a methyl functional group will replace the polar hydrogen from adenine, and we can see we can also see from the result here for adenine we have several peaks. This is because adenine, it has some isomers, so when the methyl functional group replaces the hydrogen, it have different cases. But we choose the highest peak and or target compounds when we analyze the adenine. And also we optimize the experimental conditions. Here it's the temperature for TMAH thermochemolysis. We can say with the increase of temperature, the target peak, here we choose peak 3, the peak intensity increase with the increases with the increase of temperature. And last we found 600 degrees is the optimal temperature for the detection of nucleobases. And then we tried we analyzed DA, DAMP, and DATP. They have and we know it's different. It's they enzyme function group with adenine. From their result, we found the methylated adenine is their characteristic compounds from their results. And also we found methyl phosphate and methyl furfuryl. And the important thing is we found the methylated DA here, but the peak intensity for DA, the peak intensity is the highest here. And for DA, DATP, and DMP, they are different from adenine because for adenine, we remember the 600 degree, the highest is the optimal temperature to analyze adenine. But for these 3 samples, we found actually 200 degree is their optimal temperature. And when we analyze the compounds with TMAH thermochemolysis, we found if we want to detect methyl furfuryl, the temperature should be higher than 300 degrees, but should be lower than 500 degrees. And if we want to detect the nucleosides, the temperature should be at low temperature, like from 200 degrees for 400 to 400 degrees. And here, this is the structure of poly-A tail. And when we analyze the poly-A with TMAH and SAM conditions, we also found adenine is the main compound from poly-A. But surprisingly, we also found methylated DA. Before I thought when we hit the poly-A and high temperature, for SAM, the temperature is higher to 850 degrees. And they thought that poly-A should be decomposed completely, but here we found the methyl DA structure. This means the poly-A this means the TMAH can protect the poly-A from decomposition. And last, we analyzed the real life of the E. coli. First, we can see from the results there, we found a huge amount of fatty acids, and we know fatty acids are the components of cell membrane. And the most important thing is that we found nearly all the nucleobases from E. coli, and with the detection of methyl phosphate and methyl furfuryl, we can say our TMAH thermochemolysis had the potential to detect DNA or RNA from real life. So here we give a short conclusion. We conclude that TMAH thermochemolysis could be used for the search for live biomarkers from real life. And these live biomarkers could be fatty acids, nucleobases, phosphate, and ribose derivatives. But E. coli is only the first step for our study here. And also we are doing some experiments about the bacteria that have been detected in Atacama Desert sample. And we know Atacama Desert is a Martian analog here. So all these results will provide an important reference data to interpret the data collected by rover and also get the experiment conditions of SAM and MOMA on Mars. And last, I want to thank all the fundings and thanks for all the co-authors. I'm happy to take any question. Thank you.
Thank you for the great talk. So we have a question from Anna in the chat, and she wants to know, at those high temperatures, don't you have to worry about side reactions between components?
Yes, actually, we do have for the side reactions means with our reagent TMAH or TMAH with our DNA fragments.
Anna, did you want to just unmute yourself for a second so you can clarify?
Let me check.
Sorry, I just meant any of it. Like, you know, how challenging does that make your experiments, or do you not have to worry about it very much?
You mean the temperature is high and also the side reactions, but here we do have some byproducts from our result. We can see here, but you know, when we analyze when we analyze this sample, we choose the highest peak and our target compounds, and we can see here actually the this peak is much higher than the byproducts. So
so it's usually fairly unambiguous. Yeah. Okay, great. Thank you so much.
Thank you.
And I'd like to ask, given that there are so many different temperatures that are the optimums for what you're looking for and pyrolysis is destructive, are you sort of looking to reach them all in the ramp up heat phase, or do you have to know what you're looking for before you start pyrolyzing to pick the temperature?
Sorry, I didn't follow you.
So you mentioned that, like, for nucleobases, the optimal pyrolysis temperature is 600 degrees, but for another group of organisms, it was like 300 degrees. Do you need to know in advance what you're looking for so that you can program the pyrolysis at those temperatures?
Yeah, actually we can program our pyrolyzer here. We have we can control the temperature of the pyrolyzer, and for andenine, you mean here is in 600 degree the optimal temperature, but for others it's a lower temperature like 200 degree. And I think maybe it's because you see for these three compounds we have the rebel structure and phosphate structure. Maybe when this structure help to make the detection of nuclear bases become more easier. I don't know if I answer your question or not.
That's okay. It was pretty close. I think I'll message you in the chat to ask about it later.
Thank you.
Awesome. Thank you so much for your talk. So next up, we have Alex Price, and they'll be giving a talk on microbial nitrate-dependent iron 2 oxidation as an early Martian metabolism. And I can see you're right here ready, Alex. That's great.
Yep, 1 second. Okay. Okay, can you see that?
Yes.
Great.
Yes, we can see that.
Okay. So thank you very much to the organizers for allowing me to speak at this event. It's always a lovely experience to present your work to a new audience. So that's great. So my name is Alex Price. I'm a research assistant with Astrobiology OU. That is the astrobiology research group at the Open University in the United Kingdom, where it is just past 7 a.m. But I have had coffee, so hopefully we can get through this. And I'm going to be talking to you today about some work I did during my PhD, which was about microbial nitrate-dependent Fe2 oxidation. That's a bit of a mouthful, but it's basically a microbial process that I've been investigating as a potential early Martian metabolism. So when we think about the early Martian environment, sorry. When we think about the early Martian environment and the potential for life in those environments, we really have two questions. The first of which is the habitability question. Could life have existed there? Is it possible within the parameters we know of temperature and chemical conditions, could life as we know it have existed there? And the second question is, did life exist there? This is the question of life detection. And it's one that we try to answer, we're only beginning to answer now with some of the missions that are going there. So the best place for a microbiologist like me to think about early Martian metabolisms and what could possibly have existed there is the one place we know life already exists, and that's our own planet. Now, Earth and Mars today don't look particularly similar. However, if we look at their sort of paired histories together, we can see that in the early period of Mars's history, the Noachian period, there's actually a lot of crossover between those two environments. There's a much higher impact rate on both planets from the late heavy bombardment, leading to impact-generated hydrothermal events. There are dense early anoxic atmospheres that are quite comparable in many ways. So it's there's a lot of crossover between those two early environments. Now, what kind of directions might life take on early Mars? Well, I'm mostly looking at prokaryotic single-celled life. And the reason for this becomes quite obvious if you look along the bottom of this graph, which is that complex life on Earth didn't arise for a much longer period of time. And therefore, it's reasonable to assume that if in this early Noachian period, these few 100 million years at the start of Mars's history, it was habitable, then it's likely that life didn't develop beyond a single-celled stage. Some more considerations are that evolution generally follows the environment in which life finds itself. So by thinking about energy sources, oxidants, and different carbon sources, we can begin to narrow down the types of metabolism that might have been present in an early Martian biosphere. One really good electron donor for early Mars is Fe2, that is reduced iron. Now, we all know Mars is the red planet because of Fe oxides that coat most of the surface. However, the reduced form of iron, Fe2, is there present in abundance as well, in the form of primary basaltic minerals like olivine, but also in sedimentary rocks as well, as you can see in this Mars Curiosity drill site on the right-hand side. The gray material is the reduced is from the reduced iron just beneath the surface. This is what you might think about when I'm talking about iron oxidizing microbes, these kind of acid mine drainage species that we see on Earth. However, these organisms grow using oxygen. They respire on oxygen, which is something that isn't really present in great abundance on Mars today. And we don't have much evidence to suggest that free oxygen would have been very abundant in the early Martian atmosphere either. So that's what's led me to this nitrate-dependent Fe oxidation metabolism. So as the name suggests, instead of respiring oxygen, they respire nitrate. So the electron donor is Fe2 converted to Fe3, and nitrate is reduced to nitrite and nitrogen. They live in kind of circumneutral conditions because they're very much on the sort of thermodynamic cusp. So they require circumneutral or alkaline conditions for this metabolism to be thermodynamically viable. But we do know that certain environments on early Mars were likely circumneutral based on the investigations of the Gale crater Paleo Lake by the Mars Curiosity rover. And in terms of carbon sources, we have both the CO2 atmosphere on Mars for autotrophic life, but we also have abundant organics which have been discovered more recently or confirmed more recently, again by the Curiosity rover. So how would this work hypothetically on Mars? Now this is a very busy slide, but if you look to the top left of the image, thinking about the early Martian atmosphere, it's going to be largely CO2 with some nitrogen component. Now we know that nitrates have accumulated at the Martian surface, and there have been multiple different explanations for how nitrates can be abiotically produced in the early Martian environment through thermal shock, solar irradiation, or volcanic lightning fixation. So we have nitrates accumulating at the surface. Together with that, we have active hydrology with volcanic and impact-generated hydrothermal systems, which are liberating Fe2 from the bedrock into the aqueous environments on early Mars. So you have both the electron donor and electron acceptor for this metabolism, together with the CO2 atmosphere and endogenous organics as carbon sources. So how do we actually simulate Mars in the lab? We tend to use microcosms, which are sort of small batch cultures, trying to get a little bottle of Mars, basically. And one of the first experiments I did was to look at the ability of some of these organisms to grow using a Mars-relevant mineral substrate, in this case, olivine, which is an ferrous iron silicate. So looking at the ability of these organisms to grow using only that iron from the solid olivine source as an electron donor, I found that 3 of the 4 organisms that I grew over a long duration experiment were able to increase their cell number by over an order of magnitude, and therefore grow quite well over a long period of time using only this olivine as the electron donor source, which is quite exciting. Another exciting aspect of these organisms is biomineralization, which is the production of minerals by living things. Now, under certain conditions, these NDFO microbes actually produce the Fe3 minerals, which are, so Fe3 is much less soluble than Fe2, and therefore Fe3 minerals can actually be formed in association with the cell membranes of these organisms, which is what's happening in the SEM image on the right. And so these cells can actually become encrusted in Fe3 minerals, which isn't great for the cells because they're then unable to divide. However, these encrustations encapsulate these living cells and then subsequently trap the cellular material once those cells have died. So together, these cell incrustations might act as both a morphological, but also organic and geochemical biosignature all together in one. Another tack to take is to simulate the early water chemistries of Mars, something that Michael Macey, my colleague, alluded to just before. And what I did was to use 4 Mars brines developed by Nisha Ramkissoon at the Open University and taken on by Michael, who'll be speaking later, to test the ability of my organisms to grow under simulated Martian chemical conditions. One of these is based on the Shergotite meteorites, which are representative or proposed to be representative of early Martian basalts, one of which is a hematite-rich brine based on hematite slope site on Mars. a sulfur-rich brine based on Paso Robles, and a contemporary Mars brine based on the rock nest landing site of the Curiosity rover. And you can see that they produce these beautifully colored brine media for us to grow our microorganisms in. The upshot of these experiments was that my NDFO microbes were able to grow in 2 of these 4 brines, the contemporary Mars based on rock nest and the Shergotite meteorite-based brine. Now, these are very low biomass experiments. They're autotrophic as well. So we're working very much at the lower detection limits of the biomass assay I was using, which is why you can see the large error bars on the 2 protein graphs on the right-hand side. So what I did was use multiple analyses to verify that microbial growth and metabolism was taking place. So the second set of graphs just below show the nitrite concentration. So that's the production of nitrite during nitrate reduction by these organisms throughout the experiment. And you see that it mirrors the biomass quite well. So we have some growth of these organisms in these 2 Mars brine media. One little add-on to this is that by adding an additional organic co-substrate, so no longer having autotrophic growth, but having mixotrophic growth, I guess, is beneficial to the total cell number. So you can see the blue line in the middle is when an organic co-substrate, in this case acetate, is added. And interestingly, when we add even more soluble Fe2 to that, so we have an organic co-substrate and very high levels of Fe2, you get a much greater growth benefit under conditions that usually promote cell incrustation. So the addition of organic co-substrates is an area of active research for me, and it's something I'm looking into now. So all in all, 3 strains can grow using olivine as the sole Fe2 electron donor, some of which leave mineralized structures which could potentially be used as biosignatures. Growth in, we also see growth in both the Shergotite and contemporary Mars brines, and organic co-substrates may have a very beneficial effect on growth under simulated Martian brine chemistry. And that's something I'm looking into more now. So why is all of this important specifically for active Mars research? Well, there's 2 questions that, okay, thank you. There's 2 questions I mentioned at the start of the talk come into this now, and we're moving more towards the second one with our life detection missions that are currently going to Mars. So we're trying to now answer the question of whether life did exist on Mars. So firstly, we have the NASA Perseverance rover, which is currently en route to Jezero Crater, an ancient sort of delta deposit going into a crater on Mars, which is an ideal location, a long-lived aqueous system for this kind of microbiology to take place. Similarly, we have the ExoMars Rosalind Franklin rover, hopefully landing in a couple of years' time, which is going to Oxyoplanum on the dichotomy between the Southern Highlands and the Northern Lowlands. Again, long-lived aqueous systems where these kind of metabolisms could potentially have taken place. So with that, I'd like to thank my supervisors and my research group and my PhD funders, the STFC. And as the Martian dust storm closes in on this talk, I'd like to thank you all for listening and open up to any questions. Thank you.
Thanks, Alex. So we have a question in the chat from Maggie Lau, and she said, about biomineralization, in your talk you mentioned about the deposition of iron preventing the cells from dividing. It sounded like iron deposition occurs after, during, or after the exponential phase. If not, at what phase of the cell life cycle do the iron deposits become detectable?
So the biomineralization behavior of these kind of organisms, it's not necessarily due to the stage at which the stage of the cell cycle they're in. It's more about the chemical conditions. So it's about whether or not the Fe concentrations are high enough to produce enough Fe3 that the Fe3 oxides being produced from the Fe3 being released from these organisms, whether that concentration is high enough to actually cause mineralization, both within the cell membrane, but also on the exterior of the cell, if that answers the question.
Okay, and I think I'll just quickly ask one other question. With the Martian microcosms that you were making, were they just at regular temperature, or did you change the temperature at all for those?
They were mostly at 21 degrees. So yeah.
Okay, awesome. Thank you so much for your talk and for your time.
Thank you.
So we'll be moving on now to our next round of rapid fire talks. So the talks will be one after another. So if you're asking the questions in the chat, please put an @ symbol when you ask questions, because it will actually do question times all at the end after everybody's rapid fire talk. So first up in our rapid fire talks, we're hearing from Michael Macey again, and he'll be giving a rapid fire talk on viable metabolisms in simulated Martian chemical environments.
Cool, thank you. Just checking you can hear?
Yes.
Perfect, thank you very much. Okay, so thank you for this opportunity to present again on some of the simulation experiments that we've been performing in our group. And just to be specific, when we're talking about simulating Mars, we're talking about simulating the waters that are believed to have been present on the surface during the Noachian and into the Hesperian, as evidenced by the hydrated minerals found on the surface of Mars. Now, of course, these waters wouldn't have been universal in chemistry. They would have been dependent on the local lithologies with which they interacted. So, as Alex already introduced, we are working with 4 distinct chemistries in our group based on a more general Mars chemistry, iron-enriched, sulfur-enriched, and one based on the chemistry of an early and unaltered Mars. However, in addition to the fluid chemistries that we have introduced, our colleague Dr. Nisha Ramkasoon has also developed 4 simulant regolith materials, which to date are the most chemically accurate simulant regolith materials, both in terms of elemental abundance and with regards to the Fe2 and Fe3+ ratios. Now, what we wanted to do with these, and the grand scheme for this experiment set, was to perform flow-through experiments, so more accurately simulating lacustrine environments, in order to assess whether these chemistries represented viable, habitable environments, and whether these microbes could continue growing if the nutrients were flowing through and constantly shifting as opposed to building up. However, in order to perform these experiments, we needed to select microbes with which to do this. So we, instead of using an individual strain, elected to enrich for a mixed microbial community. We did this in order to maximize the diversity of metabolisms exposed to the chemical environment, as well as to enable the community-dependent metabolisms previously mentioned. For this set of experiments, we elected to use sediment from the Colne Estuary in Essex in the UK. This was done because it's previously been characterized and shown to contain functional clades of bacteria, indicated to be relevant for the Martian environment. So sulfate
1 minute left.
Sorry?
1 minute left.
Sulfates and iron reducers and methanogens. We subbed repeatedly onto simulated fluid chemistries and the simulant material, and this resulted in the enrichment of a microbial community comprised of sulfate reducers, acetogens, heterotrophs, whilst eliminating the methanogens and halophiles originally in the system. And of course, just to show you the actual sequence data, here it is here, and you can see this shifting in bars across. So in conclusion, the simulants, fluid and materials represent habitable environments that selected for sulfate reducers and acetogens, whilst the archaea were eliminated from this enrichment series. We now need to assess how this would change under the continuous cultivation conditions, more accurately simulating lacustrine environments.
Fantastic. Thank you so much, Michael. Next up, we have Michaela Dobson, and she'll be giving an overview of the stratigraphy and preliminary results from the May 2019 drilling campaign of the 3.48 billion-year-old Dresser Formation in the Pilbara of Western Australia.
Hi, can everyone hear me and see my screen?
Yes, we can see it and hear you.
Which is the University of Auckland, and today I'm just going to talk to you about some of the work I've been doing for my PhD, which focuses on recently obtained drill core from the 3.48 billion-year-old Dresser Formation. And I would just firstly like to acknowledge the First Nation people for where my fieldwork is taking place and any other First Nation peoples present. So just a quick background for those who don't know about the Pilbara Formation. It is a geologic deposit located in the Pilbara Craton in the northwestern Australia, and is interpreted as originating from an ancient dynamic volcanic hydrothermal caldera environment. The formation consists of biosedimentary structures, including stromatolites, and recently discovered microbially mediated hot spring deposits. These hot spring deposits extended the evidence of life on land by about 500 million years. These hot spring deposits are from mapped surface outcrops that unfortunately are weathered to tens of meters depth. And because of this, a study of fresh drilled deposits was proposed. So this drilling campaign occurred in May last year, and we obtained 4 unweathered cores of about 5 to 30 meters in length. These cores show extraordinary preservation of textural biosignatures and minerals compared to the subsurface outcrops. And you can see in these photos that the core samples have sulfides preserved in the stromatolites, whereas the outcrops do not. Stratigraphic work on these cores has indicated that there's actually a high level of complexity in lithologies, which ranges on the centimeter scale in individual cores, which you can see in the stratigraphic log, to meters to hundreds of meters between cores. And here is just a couple of simplified core stratigraphy logs, and you can see that we are actually only able to correlate the stratigraphy in a couple of sections. However, when you take a step back to the kilometer scale, we actually see repetition in lithologies.
1 minute left.
Here is one of those core stratigraphy logs, and we were able to correlate it to the outcrop stratigraphy that was kilometers away from the drill site. So what was really interesting about this is that the sample that we were able to correlate was a swirly green and white silica deposit, which you can see here. We hadn't actually recognized this in outcrop before, which is really important because it shows that although the local environment was incredibly complex, there was actually repeatable successions of lithologies at regional scales. This is interesting because when we think about younger analogs that show similar associations, geothermal fields are one of these environments. The other interesting thing about this association is the only reason we identified this unit in outcrop was because the core provided us with an idea of where to expect this repeatability of lithologies by providing a 3D perspective to give a testable model, framework model of predicting where these specific lithologic successions would be present. And this is really, really important when we start thinking about exploration on other planets, because obtaining a thorough 3D perspective as well as context enables us to better predict where lithologies and possible biosignatures can be found. And just for the last minute, I just want to talk to you guys about one other deposit that we've identified in the core, which is 4 distinct spheral horizons. Petrographic analysis of these spheral horizons show interesting textures, which include internal spots, clear internal spots, fibroidal aggregates, and dumbbell shape, while Raman analysis indicates anatase-rich rims. And I am currently investigating whether these are from impact origin due to these distinct textural and mineralogy, due to this distinct texture and mineralogy. And if so, this would be the oldest identified impact spheral horizons in the geologic record. So my work that I'm currently planning to do for my PhD is to continue on with the core stratigraphy and lithologic complexity and work on those green and white silica deposits, and then undertake INAA and custom geochemical analysis on those spherals to determine if they are impact in origin. And yeah, thank you. Feel free to email me if you have any questions.
Okay, that's awesome. Thank you, Michaela. So next up in our rapid fire talks is Renee Lim talking about radicals in prebiotic chemistry.
Hi everyone, are you able to hear me?
Yes, we can hear and see you.
Okay. I'm Renee Lim, a chemistry honours student under the Albert Fahrenbach Research Group at UNSW, and I'll be talking about computing radical reactions in prebiotic chemistry. So, radical species are especially reactive, allowing the formation of a large variety of products which may otherwise not be accessible. They can also be readily generated by sources of radiation of just UV light from the sun. Radicals are hence instrumental in prebiotic synthetic scenarios, such as in the generation of starting materials and driving subsequent chemical transformations. Furthermore, the significance of radical chemistry is observed in its prevalence in various prebiotic pathways in the solid, gas, and liquid phase. For example, in the solid phase, there is protoplanetary ice grain chemistry via exposure from high-energy photons and cosmic rays, which produce radicals. In the gas phase, Miller's spark discharge on gaseous mixtures, simulating the likes of volcanic lightning scenarios, undergoes radical chemistry, and organic compounds can accumulate then in the aqueous phase. In a recent publication by the Fahrenbach group, they demonstrated how they could use radicals generated from gamma radiolysis to establish a reaction network that produces RNA precursors like 2-amino-oxazole, which is a vital precursor for pyrimidine ribonucleotide synthesis via the Pauner and Sutherland pathway. One of the key steps that is not yet well understood is the radical-initiated production of cyanamide, and this is the focus of my honors project. To investigate the production of cyanamide, we can look at the cyanamide precursor, cyanogen chloride. HCN can undergo various radical reactions to form ClCN, which in the presence of ammonia can yield cyanamide, and this is useful for pyrimidine ribonucleotide synthesis. The role of cyanogen chloride is demonstrated by radiolytic studies of briny HCN. Here, GCMS has demonstrated radiolytic yield of ClCN is increased by larger concentrations of NaCl. And here, HPLC has demonstrated that radiolytic yield of cyanamide is proportional to that of the concentration of chloride with ammonia in the solution. Some proposed mechanisms of ClCN synthesis are listed here, and in my project, I will computationally model these relevant reactions to investigate their plausibility. I use the ORCA program to perform quantum chemistry calculations in order to evaluate possible radical pathways to ClCN. These are the conditions of my calculations where the NEB method is used to estimate transition state energies. Here's a proposed reaction of HCN and a Cl radical to produce HCnCl, and I visualized this on Avogadro as well. The transition state is used to, in order to calculate the Gibbs free energy of activation.
30 seconds left.
The long-term vision is to calculate rate constants for radical reactions to piece together more complex pathways. We could evaluate the size of the reaction barriers and deduce which mechanisms are more likely to occur. And I can use high-performance computing to understand a multitude of radical reactions in the prebiotic chemistry context, which is difficult to characterize experimentally. I'd like to thank Albert, my enthusiastic supervisor, Martina Lesio of UNSW for her help with the supercomputer setup and ORCA troubleshooting, and Stephanie Vallo of the University of Washington for her help in understanding the theoretical side of quantum chemistry. Thank you so much.
Fantastic. Thank you so much for your talk. So next we have Quoc Phuong Tran giving a talk on exploring alternative starting materials for HCN-dependent pathways.
Can you hear me?
Yes, we can hear and see your presentation.
Cool. Give me one moment. Okay. So, right. My name is Quoc Phuong Tran. I'm from the Fahrenbach group, and today I will be talking to you about my work towards a self-sustaining reaction cycle from prebiotic feedstocks, and this would be under the topic of messy chemistry. Hydrogen cyanide has been shown to be a starting material for many prebiotic syntheses, such as the formation of nucleobases and amino acids. Recently, the Fahrenbach group demonstrated a precursor of ribonucleotide synthesis, namely 2-amino-oxazole, shown in red, can be generated in a continuous fashion from hydrogen cyanide using gamma radiolysis and Watt-Tricycles. In order to improve the robustness of cyanide-dependent pathways, we seek to find alternative sources of hydrogen cyanide on the early Earth. The first candidate is the amino acid glycine, which has been shown to be present on carbonaceous chondrites such as the Murchison meteorite, as well as synthesized by metallic meteorite impact and sparks discharge similar to the Yuanyuan He experiment. The second candidate is formaldehyde, formed from UV photolysis of carbon dioxide and water vapor. During our search in the literature, we came across a paper by Philip He, which describes important parameters for prebiotic network evolution, including resource availability and diversity. As shown in this figure, E and D are chemical feedstocks for the molecule A. Another parameter that caught our attention is the presence of viable cores. A core is a set of nodes in which every node is reachable from any other. And a viable core is a core that is self-sustaining given suitable food source. For example, if E is considered, if the feedstock E is available in excess, then E can generate A, which in turn produces the rest of the intermediates of this core and thus maintain the viable core. Hence, we set out to propose a reaction network inspired by this concept. Our proposed reaction network revolves around glycine, formaldehyde, and hydrogen cyanide. And it has been shown in the context of water treatment that glycine and formaldehyde can form hydrogen cyanide and then subsequently to cyanogen chloride via in the presence of monochloramine. Formaldehyde and hydrogen cyanide can come together to form glycine via the well-known Strecker synthesis, while monochloroglycine, shown here, can degrade into formaldehyde by concerted fragmentation. Formaldehyde can fuel carbohydrate synthesis by either the highly cited Formose reaction or the Kiliani-Fischer pathway, leading to simple sugars like glycoaldehyde. Cyanamide, which is formed from cyanogen chloride in the presence of ammonia, reacts with glycoaldehyde to form 2-amino oxazole, the first step of partner-silyl pathway to pyrimidine peribonucleotides.
30 seconds left.
It was reported in 1936 that glycine under ultraviolet light can form glycolic acid, which helps with peptide oligomerization by the Krishnendu group. And in 2017, they also demonstrated that glycine and formaldehyde can come together and form glyoxylate, which is the starting material for many beta-keto acid as proposed by Eschenmoser. In the lab, we have successfully demonstrated the formation of cyanamide from glycine and formaldehyde, and the next phase is trying to make monochloramine and sulfide electrons in situ. We also aim to demonstrate the continuous production of the products in intermediates and seek to understand this complex mixture better by identifying byproducts. Last but not least, I would like to acknowledge Albert, my supervisor, UNSW for supporting our research, and LC and ACA for organizing this wonderful meeting. Thank you.
Thank you so much for your rapid fire talk. That was fantastic. Okay, so now we have our last rapid fire talk for today, and that's Krishnendu Ghosh giving a talk on cancer cells as models for astrobiological study, a sneak peek to debatable insight. Okay.
Am I audible there?
Yes, so we can hear you and see you. You've just got to start the slideshow.
Sure. So thank you so much for the opportunity. And this is Krishnendu Ghosh. I'm a PhD research fellow in the Department of Zoology, University of Calcutta, and I'm working on the human brain cancer and some of its neuroimmunological aspects. And I'm going to present my thought entitled Cancer Cell as the Model for Astrobiological Study, a sneak peek to debatable insight. As we know, there are a number of theories regarding the evolution of life in the Earth. Since from the Big Bang, there are milieu of events that can actually form the life into our habitable zone. We know it is Earth. If I say this picture, this is the picture of a hydrothermal vents. Why this is important? Because presently, we all know that is the LUCA, that is our last unified common ancestor. This is the actual place where LUCA can originate. And in another context, it can be thought that can still be present, or just like the LUCA, some another thing can be present inside the icy crust of the Gallian moon of Jupiter, that is Europa. We don't know whether we can find any kind of this thing that has been shown in Avatar. We just don't know. But what we can know that the evolution of LUCA to modern eukaryotes by venture of making various kind of cellular compartmentalizations and some serious genetical modifications, some protobiological chemistry. The evolution is everywhere, because we know there are many kinds of evolution. Some are divergent, convergent, and parallel evolution. And one thing in the evolution is the evolution of cancer cell. Because we know cancer is a modern devil. It's an evolutionary disease. It is a genetical disease. It is a nutritional disease, as well as it's a lifestyle-oriented disease. So thus, it is actually being known as the emperor of all maladies. And the dismal picture regarding the cancer and its state is immense. We all know that there are multiple types of model organisms that can serve as the evolution of eukarya in astrobiological context. There are some extremophils, there are some algae, even there are some research on the fruit flies on the microgravity. We can use tardigrade, some rotifers, etc. as the astrobiological study. But can we use the cancer cell as the astrobiological study? Maybe we can, because there are lots of different nature in a cancer cell from the normal one. Actually, in course of evolution, in course of time, when a cancer cell is getting developed from a normal cell lineage, it actually becomes some sort of rebellious. And here you can see the wood reactive acetyl-CoA pathway in LUCA can be taken just as similar as the Warburg effect, by which cancer can undergo its rapid metabolism, apart from its regular glycolysis and Krebs cycle. Moreover, the differentiation of cancer stem cell from the regular cancer cell is another potential target that can be used for this kind of astrobiological study.
20 seconds left.
By taking cancer cell as the model organism, because cancer stem cells are very much tough to treat. So we can assume that cancer cell can be used as a double-edged sword. We can see the effect of space conditions. We can see the radiation on the cancer cell. And from there, we can actually devise how the eukarya develop in the prebiotic art, as well as we can use these notions as the potential therapeutic target to detect cancer in the earth. Thank you so much for the opportunity.
Okay, thank you so much for that talk. That was great. So now we have time for some general questions, just the next 8 minutes or so. I know there's been a lively debate that I couldn't keep up with about fungi in the chat, but if anybody has any specific questions for any of the presenters, it'd be great if you post them now. Otherwise, there were two general questions that I did see pop up. So one was from Kirthi, who said, I come from an ecology conservation background, and I'm not too knowledgeable about this field, but I want to know more. Since the Atacama is considered a Martian analog, how much of the area is remaining to be studied in terms of microbial diversity? Is there anybody that knows about Atacama or microbial diversity that would like to answer that question? Okay, so unfortunately, I don't know anything about microbial diversity, so I can't help with that. So I can see that there's a specific question in the chat for Michaela Dobson. So Michaela, how will you determine whether these structures that you found could be spherules or not?
So a lot of times people do, the one thing that ascertains whether they are are or not is the geochemical analysis, and that's looking at high iridium and other PGE values, as well as chromium isotopes. So I am working with ANSTO in Australia using INAA, instrument neutron activation analysis, which is one way to do this, and then the other, the CASTM system, which is with Dr. Azo in Misasa in Japan, and that's doing the chromium analysis. Can you do chromium isotope? I think so, yeah. We're still fleshing out the analytics at the moment, which is quite hard with COVID, obviously.
If you couldn't do them in situ, would you have to just like filter out the spheral? Like, how would you get them?
Maybe microdrill or do a bulk analysis and then compare it to another area of the core that doesn't have these horizons.
Cool, that's very cool and exciting. So we've just got 5 minutes left. So if anybody else has any, there's another question for you, Michaela. So what else could they be if they're not spherals?
So they could be volcanic lapilli or potentially ooids. But yeah, a lot of the texturally, they look quite promising.
Michaela, can you explain what lapilli and ooids are for the non-geologist?
Sorry. So when you have a volcanic eruption and the ash gets essentially rained on, the ash consolidates and it forms these spherical structures called lapilli, and they fall down to earth, and you can get lapilli horizons. And then carbonate ooids are just these structures that, to my knowledge, roll around at the bottom of lake beds. So they could be those, but yeah, geochemical analysis will be the true thing to determine if it is or not, which we'll probably have to wait till next year when everything opens back up.
Okay, awesome. Thanks, Michaela. Okay, so it seems like there's not any other questions coming up in the chat. So we might leave it here for today. It's been a great and long day. I know that just then being the chairperson and live tweeting everybody's talks has taken it out of me. And I'm sure that everybody is similarly tired after such a wonderful day with so much information. So thank you so much for your attention and time today. And we'll meet again at 9:55 Australian Eastern Standard Time using the same Zoom link and password tomorrow. Thank you again, everyone.